Talk about the functional classification of neurons, how neurons are categorized according to how they work or the job they do. So you've seen an example of this type of chart before, earlier in the PowerPoint. They're all the same. This one is a little bit more detailed. So as you start to learn more about neurons, you'll start to see same flow of information, same direction, but we're now able to add more information as you understand more. So you start off with the sensory receptor. Remember, the sensory receptor can be receiving information about the internal or external environment. Internal environment, what is your blood sugar level? What is your pH level? What is the temperature of a certain organ? What is the calcium level in the bloodstream, etc. External is what we generally think of as sensory receptor information: sight, sound, taste, touch, pressure, light touch, stretch. All of those are information received by the sensory receptor. The sensory receptor will then send that information to the sensory neuron. The sensory neuron is responsible for sending that information to the central nervous system so that the central nervous system can interpret it, can gather all the information it's receiving from all of the sensory neurons and put it together, process it, decide what is actually going on, then decide if you want to do something to it. So this sensory neuron is also called the afferent neuron, afferent, meaning it's going from somewhere in the periphery toward the toward the central nervous system. The interneurons enter between neurons. So these are the neurons that are between receiving the information and sending it out. Interneurons, you may have one interneuron processing the information. You may have more than one that is taking that information and sending it to the next place. So interneuron to interneuron. Then eventually, regardless of whether it's one interneuron or more than one, it's going to send information to the motor neuron. If the information from the sensory neuron, if that information is decided by the central nervous system that your body needs to do something in response, whether that is to try to increase the calcium level in the bloodstream, decrease the sugar level in the bloodstream, shiver to produce more heat, whatever that decision is, the interneuron will send that information to the motor neuron. Motor neurons will then send that information to all of the effectors that have some function in that area. So if you are cold and your body needs to or wants to start shivering, it's going to it's going to cause you to shiver in multiple different areas, both arms, probably your chest, maybe your back, your core, like several different areas will start to shiver. So it'll send that motor information to all of the neurons, to all of the muscles that need to be affected in that way. All right, so general function of neuroglia. Neuroglia will provide structural support for neurons. In the embryo, those neuroglia will help to guide the neurons into position so that it can stimulate, and it also may stimulate specialization. Instead of being a general neuron, it can then potentially have a specific function. Your neuroglia can produce growth factors that will feed or nourish the neurons, and it can also remove any excess ions and neurotransmitters from the synapse, from the synaptic cleft, from around the neuron. It can also aid in the formation of new synapses. Remember that neuroglia is nerve glue. So these are the cells that help to hold the nervous system together, support the nervous system, so the nervous system can do its job and do it effectively. Now, some of the cells of the neuroglia, you have astrocytes. Astrocytes are cells, neuroglia, that will connect the neurons to the blood vessels so that there's an exchange of nutrients and growth factors. Nerves are not necessarily going to connect directly to the bloodstream to get their own nourishment. You will have some nerves that connect directly to the bloodstream, but typically those cells are going to um, stimulate the blood vessels, so that the blood vessels can then do their job. So the astrocytes are basically the cells that will feed the neurons so that the neurons can continue to do their job. Astrocytes are going to form scar tissue anytime there's damage, anytime there's an attack, anytime there's any kind of damage the to the nerve, to the area, the astrocytes can go in um, and begin to heal those tissues, forming scar tissue. It will aid in the metabolism, in the breakdown and building up of certain substances. Astrocytes will help to regulate ion concentrations. Remember that the functioning of nerves depend on ion concentrations. You have to have a certain amount of potassium on one side of a membrane. You have to have a certain amount of sodium on one side, on the other side of the membrane. And those concentrations must be maintained at those levels in order for the nerves to be able to do their job. And the astrocytes will form part of the blood-brain barrier. The blood-brain barrier is the barrier that separates the bloodstream from the brain. Remember, the brain is very delicate. So even though your bloodstream is holding all of the nutrients that need to go from one part of the body to the other, not everything in the bloodstream is going to be compatible with the brain and the spinal cord. So the astrocytes can be a part of the barrier that will basically filter the things, the substances from the bloodstream that are trying to get into the brain and spinal cord. Sort of like the bouncer at the door of a club, not letting everybody in. Oligodendrocytes. Remember, we are talking about the neuroglia of the central nervous system. Oligodendrocytes are the cells that will myelinate the axons in the central nervous system. That is a different type of cell, or the different named cell in the peripheral nervous system. But in the central nervous system, it is oligodendrocytes. They also provide structural support for the central nervous system. So allowing the nerves to be where they are, stay where they are, help them stay supported. Microglia, these are small phagocytic cells. Remember that phagocytic cells are the ones that will gobble up and eat certain things. So you want to make sure that your central nervous system is protected. If you have debris, if you have viruses, if you have bacteria, the microglia are one of the cells that can go in, gobble it up, and hopefully remove it before it causes too much damage. The ependyma, or ependymal cells. These are cells that will line the central canal of the spinal cord. The central canal of the spinal cord is the hole that sits in the very center of the spinal cord, where you have cerebrospinal fluid flowing through there to help bathe, to help lubricate, to help nourish the spinal cord. So the ependymal cells are those that will line that central canal. It'll also line the ventricles of the brain. The ventricles of the brain are the containers, if you will, the spaces where cerebrospinal fluid flow through. And then you have the choroid plexuses. Choroid plexuses are the structures that actually produce your cerebrospinal fluid. Ependyma also help to regulate the composition of cerebrospinal fluid. So they line the structures where your cerebrospinal fluid will flow, and then they help to basically make the cerebrospinal fluid. Ependymal cells will be ciliated cuboidal or columnar cells. I know that ciliated cuboidal is not a structure that we've seen before, but do know that it can exist, and one place that it can exist is in the ependyma. So here you have a neuron. This neuron, these are the dendrites. of that neuron. This is the axon of that neuron. We're in the central nervous system. So the cells that are myelinating, that are insulating and protecting the axon of this neuron are called oligodendrocytes. You have your microglia, which are the phagocytic cells that are going to basically roam around the area, make sure that nothing is there that we don't want there. And if it is, it'll gobble it up and hopefully destroy it. You have your astrocytes, which are the cells that form your blood-brain barrier, and they help to pull nutrients out of the capillaries, the bloodstream, to feed and nourish the neuron. Then you have your ependymal cells that will line the spaces that carry your cerebrospinal fluid. In the peripheral nervous system, you have two different neuroglial cells. You have your Schwann cell. Schwann cell is the cell that actually produces the myelin sheath in the peripheral axons instead of in the central axons. Schwann cells can also speed up the speed of nerve impulse transmission. Anytime you insulate an axon, you make it such that that axon can send its signal to the next cell with increased speed. So Schwann cells will have the job of speeding up those nerve impulse transmissions because they're insulating the axons that are carrying those transmissions. Satellite cells support clusters of neurons in cell bodies. Ganglia is the name of the neuron cell body clusters that are found in the peripheral nervous system. Please make sure you look that up. You need to know what ganglia are. Satellite cells will also nourish and balance the ionic concentrations of your peripheral nervous system, the cells in your peripheral nervous system. So this chart basically makes all the cells, the neuroglia, it presents it to you in a snapshot. So you have the type of neuroglia, you have where it's located, you have the characteristics, what does it look like, and the function, what is it doing? So now let's talk about how your neuroglia help your axons to regenerate. How the neuroglia help the axons to regenerate. First thing to note, if your neurons are mature, if they are older, they will not divide. So once the neurons get to a certain age, to a certain size, they no longer reproduce. The cell is the cell, that's it. If that mature neuron cell body is injured, if the part of the neuron that has all the organelles are injured, that neuron is usually going to die. It cannot live without a cell body that is intact. And because it's neuron, because it's specialized, it will not be able to repair damage to that cell body if it's injured. So let's talk about how neurons regenerate in the peripheral nervous system. If we're in the periphery and one of the axons of the neuron is injured, it might be able to repair itself and regrow. An axon that's separated from a cell body and its myelin sheath will degenerate. So if that axon is completely separated from the cell body and the myelin sheath, that axon will degenerate. If that axon does degenerate, then the Schwann cell that is already surrounding where that axon was, it'll remain. The neurolemma will also remain. Remember, the neurolemma is what we're calling the cell membrane of a neuron. The remaining Schwann cells that were surrounding the previous axons but are still there to hopefully surround the new axon, it's going to remain and it will provide a guiding sheath for that growing axon. So as that axon starts to regenerate itself, starts to regrow, those Schwann cells that are still in place can then continue to guide that axon to where it needs to go. All right, so it can continue to follow the path of its predecessor, of the previous axon. If the growing axon does establish those former connections, if it does follow the path of the Schwann cells, then the function that you had before will return. Because the axon, the newly regenerated axon, is now touching the same structures that it did before, it will, that function that it had before will return. If it does not, if it finds a new path, then the previous function may be lost, and then there may be new function. There may be a new sensations that you didn't have before, new movement that you didn't have before. Excuse me. In your central nervous system, if in the central nervous system, the axons do not have a neurolemma. They don't have this specialized structure. So there is no neurolemma to act as a guiding sheath if that axon is destroyed. Oligodendrocytes don't proliferate if there is an injury to the oligodendrocyte. So regeneration of those axons are unlikely. In the peripheral nervous system, we had the Schwann cells that remained after injury. We had the neurolemma that remained after injury, and those structures were there to help guide the axon, the newly regenerated axon. In the central nervous system, those structures do not occur. The oligodendrocytes don't proliferate. They don't continue to divide and repair after injury. So the injured neuron does not really have anything guiding it to help it regrow. So damage in the central nervous system is much less likely to repair and regain function than damage to the periphery. So this is showing you the same thing that we just discussed. If you have a neuron here that is, in this case, this would be a motor neuron that is innervating a muscle cell fiber. If there is an injury somewhere along the axon, then the axon that is distal to the cell body, distal to the injury, that will start to degenerate, but the neurolemma and the the neurolemma and the Schwann cells will remain so that as the axon starts to regenerate, those neurolemma and Schwann cells can guide the cell, can guide the axon until the axon hopefully regains its former position so that it can regain its former function. Cell membrane potential. So a cell membrane is usually electrically charged. The electrical charge is what we're calling being polarized. So a cell membrane is usually polarized because it has an electrical charge. Having a charge difference in a particular area is called polarity. So if you have a charge difference in the axons, but maybe not necessarily in the cell body, or the polarity or the charge difference in the axons are different maybe from the cell body, then that says that those charge differences create a polarity. The inside of the cell membrane will be negatively charged with respect to the outside. So inside of that membrane, on the inside of that structure, more negative than on the outside, that is the polarity. That is what is making those cells polarized. This polarity is a result from unequal distribution of ions, meaning you have ions on both the inside and the outside of that cell membrane, but they're not going to be equal. You're not going to have the same number of ions on one side versus the other. And that unequal distribution is what is creating the polarity, allowing our cells to be electrically charged. This unequal distribution of ions is what is important so that you can conduct impulses or have electricity flow through the neurons. and through the muscle fibers. These excitable cells, cells that receive a nudge, receive a signal to do a thing, and then they can be turned on in order to do that thing. These excitable cells can rapidly change the internal charge. So we already said that the inside is more negatively charged than the outside. Once the cell gets the signal to be able to do a job, to the signal telling it that it needs to come on, it needs to do the thing, then it can rapidly change that charge on the inside of the cell. So some of those ions can move outside, some can move inside. The ability to change the charge is what allows those cells to communicate with each other. Membrane potential and distribution of ions. Membrane potential is what we're calling the charge inside the cell. The potential of the membrane potential is the ability to transport charges across the membrane. The resting membrane potential, so when a neuron is at rest, the charge on the inside, this resting membrane potential is the charge on the inside of the cell when it is not doing anything, when it is inactive, when it is just sitting down, relaxing, waiting for the next job. That charge is minus 70 millivolts. Potentials result from unequal ion distribution across the cell membrane, which we talked about previously. That is what we're calling polarity. You have potassium ions, which are indicated by K+. You have sodium ions, which are indicated by Na+. The potassium ions are higher on the inside of the cell than on the outside. So you have more potassium on the inside of the membrane. You have more sodium on the outside of the membrane. And by sequestering those potassium ions inside and having more of those sodium ions outside, that is what is creating the polarity inside the cell. That is what is creating the membrane potential. The negativity on the inside of the cell, remember the inside of the cell is more negative than the outside. That negativity inside the cell is mainly because you have a large number of charged molecules, a large number of charged particles and proteins on the inside of the cell that are impermeant. Impermeant basically means that that structure, that molecule, that protein cannot move across the membrane. It is stuck. It is stuck where it is because those membranes are selectively permeable. They only allow certain things with certain charges and certain sizes to cross the membrane. Proteins are too big to cross, so that is going to help to create the membrane potential to make that inside of the cell more negative. So not only do you have the impermeant proteins, but you also have your ions. So phosphate ion, sulfate ion, these are going to create the salts, potassium sulfate, potassium phosphate, sodium sulfate, sodium phosphate. These are going to create those salts that are going to be inside the cell. The gradients of potassium and sodium are what allow the excitability. So even though you have these ions and proteins on the inside of the cell making the inside more negative than the outside, it is the flow of the sodium and the potassium that is going to allow the cell to be excitable. Gated channels. So channels would be structures that allow certain things to go through it, just like a gate. So gated channels will allow movement of your ions, the sodium and the potassium, allow them to move in and out of the cell membrane during certain times. Not all the time, but certain times, because remember, it is the concentration of the sodium and potassium that gives the cell the membrane potential. It is also the flow of the sodium and potassium either inside or outside of the cell that allows the neuron to send the signal. So those ions are not always moving. It is just when they need to. Many chemicals and electrical factors affect opening and closing those channels. So the opening and closing of these gated channels, it's a multi-step process. Understand that it's a multi-step process, but it is because these channels are opened and closed at certain times that will allow the selective flowing of sodium and potassium in and out of the cell that create that membrane potential. On this slide, we're going to focus on action potential, what an action potential is. Please make sure that you read, excuse me, sorry about that. Please make sure that you read the rest of this information to help you understand what your action potential is. So action potential is the sequence of electrical events in an excitable cell involving changes to the membrane potential, first positive, then negative, allowing it to return to resting potential. Action potentials are used for communication between cells. So the action potential is the sequence of events in a particular cell that will allow the cell to send a signal from itself to the next cell. And what happens is you're going to have a change in the membrane potential from first being positive, then to being negative. Then it'll return to resting so that it can receive the signal, the next signal. So this would be your gated channel. The gated channel is made up, or it is a protein. You have, right now, this one is closed, so nothing can pass through. When it is time, that gate will open and that will allow the ions, whether it's potassium or sodium, to pass into or out of the cell so that that cell can pass the signal on. We're going to move past this. All right. On this slide, we'll talk about the stimulus, the threshold stimulus, and being graded. So neurons are excitable cells. Excitable cells, meaning they can receive a signal, they can receive a stimulus, and they respond to that stimulus. A stimulus is anything that can change the resting potential in either direction, either increasing or decreasing it. So a stimulus, something that can make that cell come on. It can make that cell start to function. Neurons will detect a stimulus or will detect stimuli and then respond to it by changing their resting potential. The excitatory stimulus will open the chemically gated sodium channels and sodium ions will enter the cell. So if you have an excitatory stimulus, that is going to open up the gate to allow the things to happen. That is going to change the membrane potential in that particular area. The local potential changes will be graded, meaning you'll have more in one spot than another. And then as it continues to move down that cell, you'll have more in the next spot than the previous one, more in the next spot than the previous one. So local potential changes are graded. The greater the stimulus intensity, the greater the potential change. So more stimulus can equal more potential change. An excitatory stimulus will open that chemically graded channel. This will make the inside of the neuron less negative because remember at rest, it is more negative than the outside. And then if you have a subthreshold stimulus, that won't result too much. So the threshold stimulus, how much excitement, how much movement will it take for that cell to recognize that it needs to change? If it doesn't hit that threshold, then nothing is going to happen. It has to hit that minimal threshold in order for that cell to send the signal. So subthreshold, below the threshold stimulus, there won't be an action potential that changes the cell and causes that cell to send a signal to the next cell. I'm actually going to stop there. We'll pick it up at the next video.